You can start. I think Matthews was fine to start, Shirish. So you can start. शिरीष तुम लोगों को आवाज आ रही थी मैथ्यूज की ठीक से? शिरीष,
should I start?
Just hold on, Matthews. Shirish will introduce a little bit. Yeah. I'm just asking whether they can hear us clearly or not. Shirish, can everybody hear us from the auditorium?
We can hear you, Shams. Matthews, can you hear us?
Yeah, I can hear you fine.
And what about the participants? The audio is fine. Yeah, I think we all can hear you. So I think I will begin. Let me introduce Matthews. Matthews Kokas received his BA in Chemical Engineering from National Technical University in Athens, Greece in 1994, and his PhD in Biochemical Engineering from MIT in 2000, working under the supervision of Gregory Stephanopoulos on metabolic engineering of Corynebacterium glutamicum for amino acid production. He was a visiting research scientist at DuPont Central Research in Wilmington, Delaware from 2001 to 2002, where he worked on methanotrophic bacteria for the production of high-value chemicals. He then joined the Department of Chemical and Biological Engineering at the University of Buffalo as an assistant professor in 2002 and promoted to associate professor with tenure in 2008. In 2011, he joined RPI as a career development associate professor. He was promoted to full professor in 2015 and awarded the Dorothy and Crenshaw 71 professorship in the same year. His research interests are in the area of metabolic engineering and synthetic biology, with a special emphasis on tool and strain development for the efficient production of high value and commodity chemicals. Some of his most cited works includes the development of microbial production platforms for the biosynthesis of a variety of natural products derived from plants, while recently, in collaboration with others, he has been working on developing production platforms for the biosynthesis of pharmaceutical polysaccharides. In addition, his group has worked on engineering synthetic methylotrophy, where E. coli is used to convert methanol to biofuels. He is bringing his breakthrough research to market by joining the scientific board of Chromadex, a chemical company that is currently scaling up microbial production of anthocyanins. Professor Kofas and his research groups have published 150 peer-reviewed journal publications in journals including PNAS, Nature Communications, Metabolic Engineering, Nucleic Acids Research, and others. His work, presented in the form of more than 100 invited and contributed lectures, has been featured in the National Public Radio of USA. He is fellow of AIMDE and serves as the academic editor for Biotechnology Advances, Metabolic Engineering Communications, BMC Plant Biology, and editorial board member for Metabolic Engineering, and current opinion in biotechnology. He has chaired 2 international conferences in the area of biomolecular engineering. So today I have a pleasure to invite Matthews to give a talk in this area. His talk is titled Development of Metabolic Engineering Approaches for Natural Product Synthesis. So Matthews, over to you. Thank you so much.
Great, thank you so much for the wonderful introduction, and thank you again for the invitation. It's my great pleasure to share with you some of the work that I've been doing the past several years on engineering efficient production platforms for the development of natural products. So to start with, as Shira mentioned, I work in the area of metabolic engineering. And one of the fundamental questions that we're trying to answer in metabolic engineering is what are the modifications that we need to introduce at the genome level in order to achieve what we define as cellular optimality. And for metabolic engineering purposes, cellular optimality is traditionally defined as the overproduction of chemical inputs. So in the old days, we have a target, the chemical that we're trying to produce, and you would visually inspect the metabolic network leading to the process of this particular target knowledge. And based on this digital inspection, combined with extensive biochemical data, you can come up with targets for genetic modification in order to improve the production of this particular target molecule. Now, since the mid-90s, we have developed more systematic and quantitative approaches in order to address these questions. Things such as, for example, stoichiometric modeling, functional genomics, and metabolic analysis. So either you use traditional methods or quantitative methods for combinations of the two. In the end, you identify targets for genetic modification, and you introduce those genetic modifications by using traditional recombinant DNA technology as well as more recent methods of recasted. And hopefully this will result in cell optimization or extraction of the chemical affinity. So this is a very general introduction that kind of like highlights the same questions of the structure graphs and microbes. And so based on that, today I will try to talk about the major projects that we have worked in the past 10 years in microbes. The first one is how we design phenotypes by using stoichiometric-based modeling, and specifically I will talk about a group of natural products called Flavonoid. The second part of the talk is going to be about metabolic pathway modularization and metabolic pathway balance. And finally, yes.
Your voice is not clear to us for anyone here. I don't know. Could you be a little closer to the? It's all.
Yeah, I'm actually literally stuck on my laptop right now.
Okay.
Oh yeah, is that any better? I don't know.
Maybe we'll try to stop our video. I don't know if that would work. Let me see.
Okay. All right, so I hope you can still hear me. And so this is basically the overview of the presentation. And so with that, I will start talking about stoichiometric-based modeling. So I will try to describe the overall idea behind stoichiometric-based modeling. So we start with the genome sequence of the organism that we're working with. For example, in our case, we're working a lot with E. coli, and the genome of this microbial system has been sequenced in 1997. So we convert this genome sequence into what we call a genome scale metabolic model. So every arrow that's in the figure corresponds to every single biochemical reaction that exists within the cell. And every letter that's here corresponds to intracellular metabolites that are being produced by this cellular system. So we have this genome scale metabolic map of the metabolism of the bacterium that we're working with. And then we apply very simple mass balances around every single metabolite in this genome scale metabolic map. So what does mass balance tell us is that the rate of accumulation of metabolites is equal to the rate of inflammation minus the rate of depletion. And so if you apply those simple mass balances for every single metabolite that is introduced within the cell. To give you an idea, in the case of coli, you have more than 2,000 intracellular metabolites that are being synthesized by about 1,500 different biochemicals. So this is the point where we introduce our first assumption. And the assumption that we introduce basically states that when we have this production of chemical figures, we don't have accumulation of intracellular metabolites. So what that basically means is that all the derivatives on the left-hand side of this equation they're going to become equal to 0 because, again, we don't have accumulation of intracellular metabolites. So in other words, those ordinary differential equations that we have developed here are going to be converted into simple algebraic equations. And you can represent this algebraic equations by using a matrix form. So this big matrix that you see here is what we call the stoichiometric matrix. It's basically a matrix that contains the stoichiometric coefficients of every single metabolic reaction that exists in the cell. And this vector here is a vector that contains all the rates of the metabolic reactions within the cell. So the product of the stoichiometric matrix with this vector that contains the metabolic rates is going to be equal to 0, again, because we have introduced this optimization. So if you knew some of those interstellar fluxes, you would be able to solve this system of algebraic equations by using simple algebra and quantify the unknown interstellar flux. But oftentimes you have absolutely no interstellar flux measurement. So this is the point where we introduce our second assumption. And the assumption here is that what the cell is really trying to do is not so much use the chemical features, but instead it's actually trying to grow. And this kind of makes sense from an evolutionary point of view because cells don't really care to take chemicals for us, they basically want to grow. Mathematically speaking, that translates into an optimization goal. To find out what is the optimal carbon flux distribution that will optimize the biomass generation using the constraints that we can set cells using the geometry matrix. Right. So by running this optimization, we can basically identify all the intercellular fluxes within the cell system. So now the question is, going back to the initial question, is how we can actually use this mathematical formalism in order to identify targets for genetic modification. So a few years ago, we started collaboration with state, and we can basically develop an algorithm that's called the OptiScore. So I'm going to describe very briefly what is the idea behind this algorithm. So the essence of OptiScore is finding what is the carbon flux distribution within the cell under certain constraints. So we first start by calculating what we call the raw type carbon flux distribution within the cell. And we calculate the carbon flux distribution by basically using the formalism, mathematical formalism that I kind of described earlier. So we basically optimize carbon flux distribution in order to optimize the biosynthesis of biomass. And then we run the second optimization. Only this time, we don't optimize biomass generation with what the cell is trying to do, but instead, we are optimizing for the biosynthesis of the product. So we're basically generating a new carbon flux distribution. And then we superimpose the raw type carbon flux distribution that corresponds to optimization of biomass with the carbon flux distribution that corresponds to the increased action of the chemical system. And by doing that, we can basically identify what fluxes need to be modified in order to move from the raw type phenotype to biomass phenotype into the production phenotype. So for example, this flux here basically needs to be upregulated. This flux here needs to be downregulated. This flux down here needs to be delivered. So this is kind of like how all works. And so we started applying this mathematical formalism for the production of a group of natural products that are called flavonoids. So flavonoids are polyphenolic compounds. They are phytochemicals, meaning they're compounds that are being produced from plants. And some of those flavonoids have actually been used in medicinal applications. So for example, they're now clinical trials evaluating the high-glycosylated flavonoids for diabetes treatment. However, most of the flavonoids that are currently found in the market, they're basically being nutraceuticals. Mostly because these molecules have a very strong unbalanced property. So the global market for flavonoids is more than $1 billion. So this is the pathway that highlights the flavonoid processes in plants. So you start from the aromatic amino acids such as tyrosine and alanine, and there is an enzyme called the malonyl-iase or tyrosine monoiase that results in the conversion of the aromatic amino acids into several flavonoic acids such as malic acid, basic acid, and malic acid. And then there is a lyase, ultramarine lyase, that's going to activate those 20 flavonoic acids into their CoA derivatives. So you basically form the CoA derivative of an flavonoic acid, and that's followed by perhaps the most important metabolic reaction in this metabolic pathway, a type 3 polypeptide synthase that is called chocolate synthase. And chocolate synthase basically analyzes two things. The first thing is the addition of 3 moles of malonyl-CoA, forming this linear polypeptide, followed by a cyclic concentration reaction that results in the formation of with molecules that are also substances but are fairly unstable compounds. And so once you have formed the substance, this reaction is followed by an isomerization that's called alpha isomerase, resulting in the formation of flavonoids. And once you form those flavonoids, you can basically have a number of metabolic reactions that result in the formation of more than 9,000 different flavonoids that actually exist in nature. And I keep getting messages that people are finding really very well. I'm probably not sure what else to do at this point. I'm literally fluid on laptop, so I'm pretty loud. So I hope some of you can actually follow what I'm talking about. So we basically applied, of course, in order to try and identify again metabolic pathways, metabolic reactions that need to be modified in order to overproduce the flavonoid molecules in coli. Now, one thing that I would like to highlight is the fact that in the recombinant coli strains we have developed, we don't start from the aromatic amino acids, but we start from phenylpropanoic acid derivatives, such as, for example, fumaric acid. And the reason for that is because phenylpropanoic acids are very inexpensive compounds. You can actually get them relatively easily from lignin depolymerization. So because of the fact that we're treating with phenylpropanoic acid, because of the fact that we're not treating with phenylpropanoic acid, the only metabolite that the cell needs to actually provide in this metabolic pathway is malonyl CoA. So even though, of course, it's trying to optimize the overproduction of flavonoids, what we're really optimizing for is the availability of malonyl CoA. So this metabolic map kind of highlights the targets that we map for genetic modification. So the green arrows correspond to targets for overexpression. So basically, of course, told us that we need to increase the flux to the endomeric pathway, and we also need to increase the conversion of acetyl CoA into malonyl CoA. The red arrows correspond to targets for either downregulation or deletion. So basically, of course, told us that we need to decrease the amount of acetyl CoA that enters the PCA cycle. And this kind of makes sense because acetyl CoA is the immediate precursor of malonyl CoA. Now, one thing to mention here is that back when we did this work in 2011, we didn't have very good experimental tools in order to carry out the validation. So my former student, Hank Chu, who worked on this project, ended up creating more than 40 different equalized strains based on the genetic modification targets that we derived with the culture. And to cut the story short, when we deleted two subunits for two enzymes that exist in the PCA cycle, SACT and CUP, and we combine those two deletions with overexpression of three genes, the acetyl CoA carboxylase, which is the enzyme that oxidates acetyl CoA to malonyl CoA, together with PTK and CAP-A, which are basically two enzymes that exist in the endomeric pathway. We actually created a strain that was able to produce about 390 milligrams per liter of the flavonoid molecule that were in. And to give you perspective, this is more than threefold increase compared to the production that we get from the wild type unmodified coli strain, that's only about 85 milligrams per liter. So,
Matthews, can you hear me? This is Shams. Matthews, your voice is still not very clear. Is it possible to have some other mic or something? I think it's the problem of mic most likely.
And frankly, yeah, I'm not sure how. Yeah, I don't have actually.
Okay. Okay. If there's no alternative, go ahead. Probably just try. Yeah.
I'm literally on my laptop, but I turned off my camera. I can't.
Yeah, it's not very clear what you are saying, so somehow.
I mean, do you want to continue? Do you want to stop? I don't know.
Because we can't hear you very clearly.
Yeah. 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 I mean, I don't know. I don't know. I don't know what to do. I mean, I'm not hearing that much to the point. I don't know.
Okay, you go ahead. I guess there's no alternative. Then you go ahead and just be a little slow and near to the mic.
I don't know how it all.
Go ahead. Go ahead. Thank you.
Okay. So for those of you who here, the next thing that we want to see is whether we can actually use those genetic modifications to test the overproduction of other chemicals that are specified from our way. And we always target to do about this kind of molecules is fatty acids. So this is basically metabolic and one fatty acid in coli. So in coli, in order to produce fatty acids from glucose, go to the enzyme, not pathway, all the way acetyl way, and then again we have the acetyl phosphate acetyl way in pathway. And once we form our little way, we have the fatty acid of these pathways to be so with a condensated carboxylation reaction that's followed by a reduction and ketone hydrate, followed by another reduction. And every time we go through this fatty acid pathway, we basically consume one mole of polymer way. And then you have a group of enzymes, Inoculation, that are called bioenergy, that release the free fatty acids from the acetyl ACE receptor. In E. coli, we also have a beta oxidation pathway that basically converts those free fatty acids into their proteases, going all the way back to the acetyl way. So when we wanted to test the applicability of some of those genetic modifications in fatty acid production, the very first thing that we did was with fat feed. So fat feed is the very first step in the beta oxidation pathway, and this is work that others have actually done before. So when we delivered fat feed, we really see a significant increase in the production of free fatty acids. We then combined the deletion of fat feed with overexpression of the acetyl CoA carboxylate. And so when we did that, we actually saw a significant increase in production, so the titer jumped to about 300 milligrams per liter. And the next thing that we did was combine the deletion of fat feed with overexpression of acetyl CoA carboxylate, which by the way is a fourth-amino enzyme in E. coli, and the overexpression of E. coli bioesterase, which again is the enzyme that produces free fatty acids from the acetyl ACE precursor. And that resulted in an additional increase in production, so the production jumped to about 600 milligrams per liter. We also tested the overexpression of other biosterases are basically derived from plants that have been reported in the literature to have increased availability of fatty acids. So when we introduced those recombinant biosterases, we didn't really see an increase in the final titer of the free fatty acids. What we saw, however, was that the type of free fatty acids produced changed. So when we overexpress the E. coli bioesterase, we produce mostly C13-saturated free fatty acids. When we overexpress the plant biosterases, we produce mostly C16 saturated free fatty acids. And then we started adding some of the genetic modifications that we identified with, of course, for improvement of Flavonoid Pathway. So the first thing that we did was we overexpressed the two glycolytic enzymes, CPK and GAPA, and we did that by using high copy number plasmid and a medium copy number plasmid. So when we introduced those two overexpressions, we saw a slight increase in the production. It jumped about 750 milligrams per liter. We also overexpressed 4 genes that are involved in the fatty acid aggregation pathway, again by using a medium copy number plasmid and a high copy number plasmid. So by doing that, we increased the final titer to about 1 gram per liter of free fatty acids. And the final thing that we did was combine all the genetic modifications. So the deletion of the fat D, the overexpression of the acetyl-CoA carboxylase, the overexpression of the diasterase, the overexpression of the glycolytic enzymes, as well as the overexpression of 4 fatty acid allocation pathways. And in this case, not only we didn't see a further increase in production, so the production actually dropped to about 800 milligrams per liter. So the hypothesis that we developed based on these results is perhaps in the case of free fatty acids, not only we need to identify the genetic modifications that we need to introduce to improve the titer, similar to what we did with the flavonoids, we also need to take into consideration the fact that fatty acid biosynthesis is a very long metabolic pathway. So in other words, we actually need to balance this metabolic pathway. So fatty acid biosynthesis starting from glucose involves more than 14 metabolic steps in E. coli. So we took this very long metabolic pathway and we broke it down into 3 metabolic modules. The first module is what we call the glycolysis module. The second module is what we call the acetyl-CoA activation module, which is basically the 4 subunits of acetyl-CoA carboxylase. And the third module is the fatty acid elongation module, which basically involves 4 enzymes that catalyze the fatty acid elongation pathway plus the diasterase. And each one of those modules was overexpressed in the form of an operant. So what that basically means is that the genes that are involved in these modules were all expressed by using one single promoter. And each one of those operants was introduced again in high copy number plasmid, medium copy number plasmid, and a low copy number plasmid. And we tested different combinations. And we started to cut the story short. When we overexpress the glycolytic module that basically carries the ENT pathway overexpressions on a medium copy number plasmid, the acetyl-CoA carboxylase, the 4 subunits of the acetyl-CoA carboxylase on a low copy number plasmid, and the fatty acid synthesis module on a high copy number plasmid, we actually achieved a final titer of 1.4 grams per liter. So in this case, we actually did see an increase in the production. And so we moved this strain into a fed-based fermenter, and the final titer that we achieved was 8.6 grams per liter, which is not the highest production of free fatty acids that has been reported in the literature. So people have actually reported 15 grams per liter. However, the production rate of 0.124 grams per liter per hour was the highest production rate that had been reported in the literature at that time. All right, so that basically is an example. The fatty acid biosynthesis is an example of the fact that oftentimes not only do we take into consideration the metabolic modifications that we need to introduce in order to overexpress a particular metabolite, but oftentimes we actually need to balance the metabolic pathway. So keeping on this topic, so we decided to basically move this concept of metabolic pathway balancing a step further. So instead of actually using different plasmids with different copy numbers in order to balance metabolic pathways, we basically decided to split a long metabolic pathway into 2 different microbial strains. So the kind of case study that I highlight in this topic involves the biosynthesis of a natural product that's called flavonoids, commonly known as green tea catechins. So green tea catechins are aromatic compounds that are also synthesized from 10 propanoic acids. So you start from fumaric acid. Again, you go through a 3-step pathway that involves the 4-fumaryl ligase, sulfone synthase, and sulfone isomerase, resulting in the formation of the flavonoids. And then there is the 3-step pathway that involves the hydroxylation, coupled with 2 reduction reactions, hydroflavonol reductase and glucosamine reductase, that both require an ATPase that result in the formation of the green tea catechins. So this is a 6-step pathway, and what we decided to do was take the first module of this pathway that involves 3 enzymes, 4-CL, CHS, and CHI, and overexpress this recombinant pathway in the E. coli strain that often produces malonyl-CoA, because malonyl-CoA is the precursor in this three-step pathway. And then the other three enzymes, the FBAs and the two reductases that basically require NADPH, introduce them into another E. coli strain that has basically been engineered to overproduce NADPH. In other words, it has been engineered to send most of the carbon to the pentose phosphate pathway. And then we mix the two strains together. So we created what we call an artificial co-culture. And in order to basically optimize this artificial co-culture, we tested 4 different parameters. The first one is what we call the inoculation ratio. So how much of the first strain and how much of the second strain do you inoculate at time point 0 when you start the co-culture. The second parameter is the induction time. So at which point, after you start the co-culture, you add the inducer of the recombinant pathway. So in our case, we're basically using IPG to induce the recombinant pathway. The third parameter is the carbon source. So everything that I presented so far requires the use of glucose. We also tested the use of glycerol as an alternative carbon source, as well as mixtures of glucose and glycerol. And the final parameter, the fourth parameter, is the temperature. So as you all know, E. coli precursor growth at 7 degrees. We also tested lower temperatures, so we went all the way down to 10 Celsius. And the results of those optimization experiments, we used them in order to basically identify the optimum fermentation parameters using a 4th-order scale Gaussian model. So this is a mathematical work that was not actually done in my lab, but in a collaborative lab. But the bottom line is that this 4th-order Gaussian model allowed us to identify what is the optimum inoculation ratio, what is the optimum temperature, what is the optimum carbon source, and what is the optimum induction point in order to optimize the biosynthesis of the flavonoids using this co-culture approach. And so it resulted in a production of 41 milligrams per liter of green tea catechins using this co-culture approach, which is definitely not the highest production rate that you can imagine. However, it's a 58-fold improvement compared to the production you get from a consolidated process. So if you basically take all 6 metabolic pathways and you put them into one single E. coli strain. And then we further extended this approach of co-culture by using a 4-strain co-culture that we call polyculture in order to basically synthesize another group of natural products that are called anthocyanins, again starting from glucose. So here we're using 4 strains. The first strain has been optimized for the biosynthesis of phenylpropanoic acids. The second strain has been optimized for converting these phenylpropanoic acids into flavonoids. The third strain takes those flavonoids, convert them into the green tea catechins. And finally, those green tea catechins are used by the 4th strain in order to basically be converted into anthocyanins, which is a natural pigment that is being explored now in food applications. So this is basically a case study that highlights the concept again of metabolic pathway balancing and how oftentimes we need to use to balance metabolic pathways in order to achieve improvement in final patterns. So the last thing that I'll talk about, and I apologize again that you can't hear me very well, I keep seeing more comments about this. The last thing that I will talk about is the biosynthesis of another group of natural products that are called sulfated glycosaminoglycans. So glycosaminoglycans are polysaccharides that are animal derived, and they are basically used in pharmaceutical and nutraceutical applications. So the most famous of those polysaccharides is a glycosaminoglycan that's called heparin. So heparin is the most commonly used anticoagulant drug. Another one is chondroitin sulfate. This is a sulfated glycosaminoglycan that is used as a nutraceutical for people that suffer from joint health issues and osteoarthritis. And it's actually also being used for drug delivery applications. So all sulfated glycosaminoglycans are animal-derived polysaccharides. The majority of those polysaccharides is being produced in China, so more than 50% of the global supply of sulfated glycosaminoglycans originates in China, and then you have production in other places such as the European Union, the US, Brazil, and Canada. So there are several issues with the current production method that involves extraction from animal sources. So one problem is that there is a lot of variability in the chemistry of the polysaccharide we are going to produce, depending on the animal source, the animal that we're going to use to extract those polysaccharides, and also the tissue that we're going to use. They're also very prone to contamination, as something that I'll touch upon in a couple of slides. Contamination by viruses, animal viruses, as well as fear. And finally, there is also a lot of concerns nowadays about ethical treatment of animals, and also religious concerns, especially for animal-derived products coming from pigs. So the first of those sulfated carboxyluminoglycans that I'll be talking about is heparin, which, as I mentioned to you, is an anticoagulant drug. So heparin, again, is sulfated carboxyluminoglycan. So you have a polysaccharide that is being composed by undersulfated disaccharide repeating units that are interspersed with trisulfated disaccharide repeating units. And the result of this trisaccharide repeating unit would be trisulfated repeating unit with undersulfated repeating unit is the formation of this pentasaccharide binding site. This is basically where antithrombin is going to bind to the heparin, and it's going to basically exercise its anticoagulant properties. So this is the facility for extracting heparin in a slaughterhouse in Jiangsu province in China. So you basically get about 300 milligrams of heparin per pig. And so this is what we call the raw heparin that we can basically buy for about $100 per kilo. And this is what gets exported from China across the world. And this raw heparin is then going to undergo extensive downstream processing that involves a lot of column purification, resulting in what we know as pharmaceutical grade heparin, which is what's used as anticoagulant, that is commonly sold for about $10,000 per kilo. So there is more than 1,000-fold increase in the value of the molecule when you go from raw heparin all the way to pharmaceutical heparin. So back in 2007, 2008, there was a big contamination crisis. So what was supposed to be raw heparin being exported from China, it ended up being trisulfate, polysulfated, as we say, chondroitin. And so the result of that was that more than 100 people died in the US, and many other people died across the world as a result of this contamination. So back in 2011, we started a project on using metabolic engineering in order to produce animal-free heparin, and this is the process that we have actually developed. So we start with an E. coli strain that is called K5. So this E. coli strain produces an unsulfated polysaccharide that kind of looks like heparin. So the first step that we need to do is an alkali treatment in order to reduce the molecular weight of this polysaccharide. And this is followed by N-deacetylation using chemical catalysis. And then that's followed by the sulfation of the three amino groups that are generated once you carry out the deacetylation. So this N-sulfation is also carried out by using chemical catalysis. And once you have formed this intermediate, this N-sulfa N-acetyl comparison, we basically carry out 4 sequential enzymatic modifications to this polysaccharide. So we have an dimerization that's converting glucuronic acid into alduronic acid, and that's followed by 3 sulfation reactions that result in the formation of a sulfated polysaccharide that basically resembles the animal-derived heparin. And so in 2019, we basically published a paper in Academic League where we basically demonstrated for the first time the use of this chemoenzymatic method in order to produce anticoagulant heparin, basically heparin that has the anticoagulant properties of the animal-derived heparin. And not only that, we actually use this chemoenzymatic method in order to actually develop a deuterated version of heparin, something that basically demonstrates that you can actually use this chemoenzymatic method to actually make different types of derivatives of heparin, potentially that have better anticoagulant pharmaceutical properties. So we have also worked with another sulfated glycosaminoglycan that's called chondroitin sulfate. So chondroitin sulfate is also a sulfated polysaccharide that derives from animal sources, from bovine trachea, but also from shark fins. And so the story is pretty much the same. We're basically trying to use metabolic engineering in order to basically produce an animal-free version of chondroitin sulfate. So back in 2014, we isolated an E. coli strain, a neuropathogenic E. coli strain that's called E. coli K4. And this E. coli strain can actually produce a polysaccharide, an unsulfated polysaccharide that does look like chondroitin. The only problem is that this polysaccharide has this beta-fructosyl group that's branching out from the main polysaccharide chain. So we sequenced this K4 E. coli strain, and we identified 3 gene clusters that are responsible for the biosynthesis of the polysaccharide, as well as its transport to the cell wall of the E. coli K4 strain. It turns out you only need 3 genes, KFOF, KFOC, and KFOA, in order to be able to make an unfructosylated version of this polysaccharide. And we developed a PL21 strain. So K4 is a neuropathogenic strain, so you cannot actually use this K4 strain in a fermentation. So we actually express those 3 genes in E. coli, PL21, which is basically a safe E. coli strain. And we're able to basically produce in a chemostat about 2.4 grams per liter of the unsulfated chondroitin. Now, the question now is how we can actually make a sulfated chondroitin by using E. coli. And so in order to be able to do that, we basically followed a metabolic engineering strategy that focuses on 3 different things. The first one is deletion of a fructosyl transferase gene in E. coli K4 in order to basically remove the beta-fructosyl group that branches out from the polysaccharide gene. The second thing that needs to happen is the overexpression of the sulfur transferases, the enzymes that basically are responsible for sulfating the polysaccharide. And the third component is the biosynthesis of PAPS. So PAPS is an ATP derivative. It's basically the sulfur donor for the majority of sulfation reactions that happen within E. coli. So in order to basically overexpress the availability of PAPS in E. coli K4, we started by overexpressing a number of biosynthetic genes that are involved in the biosynthesis of PAPS. So when we did that, we didn't really see any significant improvement in PAPS availability. The next thing that we tested was the downregulation or potentially deletion of this gene that I highlight here in red, that's called cis8. It basically encodes for PAPS reductase, and this is the only enzyme that exists in E. coli that can actually degrade PAPS. So initially we wanted to delete this enzyme. We were not able to delete this particular enzyme, so we used CRISPR-I in order to repress the availability of cis8. And when we did that, we were actually able to show for the first time the biosynthesis of sulfated chondroitin when you also overexpress chondroitin sulfur transferases in this recombinant E. coli K4 strain. So the sulfation percentage was extremely low, so less than 10% of the polysaccharide that we actually produced was actually sulfated. And finally, when we were able to finally delete the cis, that gave us a further increase in the percentage of sulfation, so we got about 19% of the chondroitin that we produced to be in sulfation state. In terms of production titer, we were able to produce about 120 micrograms of chondroitin sulfate per gram of dry cell weight. So all the chondroitin sulfate that we actually produce is intracellular. So we wanted to see if we can actually further increase the sulfation percentage, because the sulfation percentage is actually the most critical chemical component that determines the activity of chondroitin sulfate. So we were able to basically show that chondroitin sulfur transferases are not enzymes that express very well in E. coli, and there are several reasons for that. So chondroitin sulfur transferases are enzymes that reside in the Golgi of the cell systems that actually produce chondroitin sulfate, and they're glycoproteins. So E. coli doesn't have Golgi, and of course you cannot glycosylate those enzymes. So we wanted to basically engineer the sulfur transferases in order to improve their functional expression in E. coli. So the very first thing that we did was we removed the N-terminus of the chondroitin sulfur transferases that's basically responsible for attaching those enzymes in the Golgi apparatus. And then we were able to slightly improve the activity of those enzymes by introducing a number of different tags, such as the TRX8 tag and the 6-histidine tag. So those tags were basically able to improve the solubility of this enzyme, but still the activity that we're getting is extremely low. So a former postdoc in my group, Dr. Patrick Fraser, and a former graduate student, Asher Williams, they basically used a computational tool that's called PROS. It's a computational tool that's publicly available. And what PROS allows you to do, it allows you to identify mutations that basically improve the stability and the solubility of an enzyme when you express it in E. coli. So we were able to actually develop a number of different mutants based sulfur transferase, chondroitin sulfur transferase mutants based on the predictions that came out from PROS. And to cut the story short, we're able to show that all three of those mutants, I call them SM1, SM2, and SM4, actually showed substantially higher enzymatic activity compared to the wild-type activity when we express them in E. coli. And when we introduced those mutant sulfur transferases in recombinant E. coli, we were able to actually improve the sulfation percentage quite substantially compared to the sulfation that we get from the unmutated wild-type sulfur transferases. I know I'm running out of time. I'm just going to mention one last thing. One of the things that we've noticed during this work is that all the chondroitin sulfate was intracellular. And chondroitin that was actually secreted in the medium was unsulfated chondroitin. So we developed the hypothesis that there is a competition between the transport of the chondroitin to the extracellular medium and sulfation of chondroitin within the cell. If sulfation happens, then chondroitin sulfate gets trapped. For whatever reason, it cannot get secreted from the cell. If, however, it doesn't happen, then chondroitin, the unsulfated chondroitin, is basically going to get secreted from E. coli. So what we did was we introduced CRISPR-I in order to downregulate a couple of enzymes, KPSM and KPST, two enzymes that are involved in the transport of chondroitin to the extracellular medium. And when we reduce the expression of those enzymes using CRISPR-I, we actually show a significant increase in the percentage of sulfation of the chondroitin that we're making. About 55% of the chondroitin that we're producing was actually sulfated chondroitin. And this basically map kind of shows the sulfation percentage of chondroitins derived from different animal sources. So starch cartilates, when you use starch cartilates to produce chondroitin sulfate, you have about 83% of the chondroitin being sulfated. When you use whale cartilates or bovine trachea, the percentage sulfation is between 73 and 69. And again, the E. coli strains that we have developed give us a sulfation percentage between 55 and 57%. So we're currently actually testing some additional sulfur transferase mutants in order to further increase the sulfation percentage of the chondroitin we're making. Again, the sulfation percentage is perhaps the most critical thing in determining the biological activities of those molecules. So I would just like to acknowledge my collaborator, Paul Pinkford, who has actually helped me tremendously with the analytical work involved in those projects that I discussed today. And I would definitely like to acknowledge funding from several different sources: the National Science Foundation, the National Institute of Health, and a number of private companies that have funded different aspects of the projects that I discussed today. Again, my apologies, you can't hear me very well. If you have any questions, I'll be happy to answer them.
Thank you, Matthias, for the wonderful talk. We are open for questions. So any questions from our online audience? You can speak up your question as well, the online audience. So, any questions from the offline audience who are physically present here? So, I mean, maybe I'll get started. Very interesting work, Matthias. In terms of a couple of things, so for example, when we are talking about, let's say here we are focusing a little bit on bioenergy production and then engineering various strains. So you have shown an example of fatty acid based molecule production. So in terms of that, obviously their reducing power plays a major role. So what are the common approaches, and I guess this is also for all the audience that are present here, in terms of because E. coli has a little bit more preference towards NADH versus for fatty acid and their derivative production you would have to use NADPH. So how much of a struggle that is in terms of making E. coli like or produce more of NADPH?
Yeah, so that's a great question. So most of the work that we have done on fatty acids has focused on malonyl CoA availability. We did, however, do some work for improving NADPH. And NADPH in E. coli is being produced, among other things, by channeling more flux through the pentose phosphate pathway, right? So the pentose phosphate basically gives you NADPH. And this is basically a paper that we published in 2015. We did use OptForce. We also used another computational model that we developed in-house in order to identify knockouts that improved NADPH availability. And all the knockouts that were predicted basically meant that we would have to shut down flux through the Enter-Markov-Parnasse pathway and send most of the flux through the pentose phosphate pathway. And that's exactly what we did. So we basically, the best strain that gave us the best accumulation of NADPH involved the knockout of PGI, phosphoglucose, phosphofructokinase, I'm sorry, the knockout of phosphofructokinase, and the overexpression of ZWF, which is basically the very first step in the pentose phosphate pathway. So we published this paper in 2015, and that actually, we used this strain in order to produce a group of natural products that are called the green tea catechins that I actually mentioned briefly in my talk today. So that's the metabolic engineering strategy that worked very well in our hands for NADPH.
Right. And I mean, I do not know what you just did because your sound quality just skyrocketed. I mean, we can hear you now perfectly crystal clear. Anyway,
You can hear me now, okay?
perfectly better now that your slideshow is off.
I have no idea. I don't know what to say.
Yeah, no issues. So I mean, I guess so again, another opportunity for our online participants to ask questions. So those of you who are interested in online, they can raise their hand if there are multiple, then we can ask them in order. So again, I guess I'll have another follow-up question, I guess, in the meanwhile. So in terms of choice of E. coli strains, I know for your chondroitin sulfate work, you had choose BL21 for this work. Any particular reason why you chose this strain versus the other available strains?
So we use the T7 promoter a lot in my lab, and that was pretty much the only motivation for using BL21. Now, having said that, when we tried, so we used BL21 to make a non-sulfated chondroitin. We didn't really try any other E. coli strains, so we got it to about 2.4 grams per liter. Now, when we tried to overexpress the sulfotransferases, in BL21, in order to get sulfated chondroitin, which is what we really need to produce, we could get zero activity. There were only very specific E. coli strains, such as, for example, the origami E. coli strain that actually gave us some activity of the sulfotransferase. And of course, part of the reason I think is because there are a lot of disulfide bonds in these enzymes. So eventually, we ended up switching back to E. coli K4 in order to basically produce the sulfated version of chondroitin. And that was basically the E. coli strain that gave us the best production. We did try one more E. coli strain, the MG1615, if I remember correctly, but we got some chondroitin sulfate, but not as much as what we got out of K4.
Right, right. Awesome. Yeah, I mean, and again, I guess this is a perfect example of the background struggle experimental that we need to do. Modeling can take us a little bit where we want to go, but then all of these are practical problems that one needs to overcome. So I guess a last call for a question from either online or offline participants. Okay, now finally we have got one raised hand. Go ahead. One second. Hello. Yeah.
Hello, sir.
Hello.
Hello. So you mentioned about the NADPH flux. So you overexpress the PFK gene towards
No, he deleted the
he deleted.
and overexpressed ZWF, which is the first enzyme in the PPP.
Right.
Okay. Then what about the further glycolysis pathway?
So yeah.
Mm-hmm. Yeah, so we actually did try a couple of knockouts in the glycolytic path. So I guess when you say glycolysis, you mean the anti-meric pathway, right? So we did try a couple of deletions in the AMP pathway. I don't remember exactly. I want to say it was the transketolase, which is one of the genes that connect the pentose phosphate with the AMP pathway. Not only we didn't see any improvement, actually the growth rate dropped quite substantially when we tried some of those deletions. So the phosphofructokinase deletion was the one that worked the best in the AMP pathway.
Excellent. So and again, another example of just to achieve one thing, you may have to try 3-4 different approaches for one particular goal. So excellent. And again, for all the online as well as offline audience, I think I must also tell that Matthias is joining us today from Hong Kong, so he is traveling and he has taken out time to present his work to you, and we really sincerely appreciate you taking the time. And I guess obviously we did benefit from your talk, so thank you, Matthias, for taking time today.
Thank you so much, and again I apologize for the bad sound. I wish I could do something more.
Yeah, no, thank you, Matthew. I think your last part of the talk, almost last 30-40% was fine. Only the first half was really bad. So,
Okay.
I guess the interesting part was still okay to me. Okay, great. So thank you very much. Thanks from my side as well, Matthew. It was a pleasure seeing you again. Have a good day. Bye-bye.